Thank you for having us. So Seshu and I are really going to try to just give you a flavor today of what we're doing with artificial intelligence for manufacturing for cell and gene therapies, just to kind of help you see how we're thinking about the problem, what kinds of problems we're solving with what kinds of data, and open up the discussion. So let's get to the next slide. Just a brief disclaimer on today's presentation that you all saw. Okay, so I really like to start all this whenever people talk about AI, this apply to whatever area you could think about, right? Whether it's build a disease model from omic data or apply it to understanding the manufacturing process and supply chain processes to healthcare, right? And medicine specifically is why do we think this is possible now? And for me, it really starts with the data, right? Our now unprecedented ability to collect all kinds of measures, both on the patient level, whether we're talking about something as granular as your genome or molecular readout or phenotypic screen readout, or actual health outcomes in a single point in time and over time, but also things that we might be learning about, how we go about generating and creating an effective cell therapy or a new drug. So all of that is producing lots of data where we now have the potential to learn from that data. And you combine that with advances in AI. So from the AI side, it's really the realization that we can now come up with algorithms, deep learning algorithms just being a class of algorithms on the AI side that we can use and apply and leverage that can learn the complex patterns underlying the data. And the more data you feed it, the more accurate these algorithms could potentially get, exceeding what we were able to do 5-10 years ago with machine learning, and combining that really with then what we're able to do from the computational power side. And hopefully all of these things together improve our ability overall to learn from our data, predict over our data, and reason over our data. So if we go to the next slide. And just as evidence in terms of what AI can do in our field. So diabetic macular degeneration, and as it relates to other follow-on diseases like diabetic retinopathy and whatnot, we've seen how we can start from retinal images, use high-performance supercomputing along with AI to get to now outcomes that look interesting to us. We can actually learn things over the data that we couldn't before, right? Starting with the ability to get to algorithms that can exceed what an ophthalmologist could do, to now finding new associations that go beyond the imaging data, combined with biomarker data, so we can learn about how all of these things potentially from macular degeneration could impact things like adverse cardiac events, and even getting to an algorithm that could be recognized by the FDA and get actual FDA approval so that we can start to apply it at the point of care. So this is sort of the possibilities of us knowing that there is potential here with AI and data and what we can do with it. And we're starting to see the fruits of that emerge. Let's go to the next slide. All right, so what are we doing at Novartis? Really our view is, can we harness core AI capabilities and advances in AI capabilities that not just look at deep learning, but even beyond deep learning, various approaches to doing causal learning along with deep learning, various approaches to harnessing data coming from text and files, such as NLP analytics, and approaches to doing imaging where we can take readouts that we will see in the manufacturing process, such as flow data. How could we leverage that and apply AI to automating our analysis of that so that it doesn't become a tedious job for the human, and combined with advances in visual analytics. So we're not just looking at sort of one deep AI field, but really the combinations of these capabilities, building them out, and then applying them in two different ways. One is to really understand what we call exploration, the fundamental mechanisms and processes that drive what we're seeing, that drive the outcomes that we're trying to model. And in the second, coming up with capabilities that empower many people. So not just the deep data AI scientist that knows how to leverage these tools, but also a data scientist, even a non-data scientist that wants to be able to interact better with the data and reason with it. So we're doing both of these things, and we think it's important to really being able to harness the potential and power of AI, not only to do fundamental discoveries, but also empower our scientists and our clinicians throughout the organization. Next slide. All right, so just an example of some of what we're doing in visual analytics applied to flow cytometry data. Next slide. Okay, and we're not just looking at things like difficult data sources to analyze, like imaging, but also how do we gain insights from data that's really unstructured, massive amounts of corpus text data, applying NLP capabilities along with knowledge graph capabilities to better analyze that. Next slide. Okay, looking at things like how do we better extract insights from text data to automate our ability to learn and extract adverse FDA events and apply it to our drug labels. Next slide. Just highlights and previews here. To better ways to predict outcomes using EHR data, right? So right now, if we want to gain insights from our EHRs, we have to both really deploy a lot of people to pull out what we can from the combination of the NLP records along with the EHR records, and various AI and NLP methods can help us kind of do that at scale and very quickly. Next slide. And another fundamental capability that we're harnessing is, and we'll see this later in terms of what we're doing specifically in cell and gene, is being able to learn cause and effect from data. And so there's, one thinking is that we're going to learn these patterns. The patterns are complex, and in some way between your inputs and your outputs, it's a bit of a black box, but let the machine kind of figure out what the inner workings is. But another is we actually want transparency into what underlies the system. What is the process and system that gave rise to the data? And so through causal learning and graphical causal learning methods, we can take these data sets and start to learn, given a set of inputs that are modulating the response in the system, how does that lead to a potential causal network that we could learn and predict that would tell us how it's influencing the outcomes that we care about, and whether those nodes that we're measuring are causal for the outcome or reactive to the outcome. If we go to the next slide. And we want to be able to do the sorting of causality because it fundamentally helps us deal with confounding factors, which is a big deal and a big underlying issue with these data sets. Because if you're just trying to look at patterns and large-scale patterns of association, you would incorrectly infer that amputation and insulin are just somehow associated with it, when we know that there's an underlying cause, in this case, a confounding factor that says if you have diabetes, that will influence your changes in insulin, which could influence amputation, and then diabetes directly to amputation. And what's not shown very well are the underlying statistical and mathematical probabilities that allow us to sort out cause and effect. And so we can do this now mathematically, but if we go to the next slide, we're also able to take this, working with various partners. This is my previous company where we developed technology for not only learning cause and effect associations at a small graphical level, but being able to do it at scale. We go to the next slide. Okay, so this is all sort of the intro to what we are doing today in cell and gene, which is really to highlight that we're leveraging several capabilities from the AI field across different data modalities and different types that allow us to deal with imaging data, NLP data, allow us to sort out cause and effect, because there's huge potential to hopefully start to learn what are the various key steps in this manufacturing process, starting with the patient to getting the treatment and the cell therapy into the patient and being able to monitor them long-term, collecting all kinds of data modalities along the way. Can we now put this all together and leverage various AI capabilities to really learn and understand the process? And also then, and why we touched upon cause and effect, be able to intervene and figure out where do we need to intervene to optimize that. And hopefully our AI and machine learning models will enable us to do that. And so with that, Seshu will talk about what we're doing in the cell and gene field specifically.
Thank you, Aya. In Cell and Gene, our long-term vision and mission is the end-to-end patient journey. How can we understand this as best as we can with the goal of improving every single step through the way, such that improving incoming material, which can then help us in better patient selection, which would then lead us to improved and robust manufacturing. This would then lead us to reduce vein-to-vein time, improve predictability, and in the end, it'll allow us to reach a greater patient population. Obviously, cell and gene is a very growing field, very nascent field, so there are a lot of challenges. I've just listed some of the challenges here. The biggest ones being there is tremendous donor-to-donor variability and process variability because we're still learning the process as we go. Analytical assays, vendors, logistics, trained personal and rapid and aggressive timelines. So anything we can do to learn, to understand, so we can expedite or remove an obstacle is always helpful. This being the mission, I think I'm going to skip over the next slide in the interest of time. This is basically talking about autologous CAR T cell therapy, which I am assuming that most of you at least have an idea. We start with the patient, we modify their T cells, and then expand them in-house, and then formulate, freeze, and then send it back to the patient, and the patient is infused. So the next slide here is actually taking that data set that I just mentioned and breaking it down into where are the pieces of data, points where we are collecting the data, much in line with what Aya had mentioned in the complex picture earlier. So we have the patient, we have apheresis, manufacturing, final product, cellular kinetics, and other characteristics. As we mentioned just now, there's a cause and effect, and each of these has an impact on the next step or a few other next steps. So each stage affects the subsequent, and there's dependencies. And the data sets that we are able to collect at each of these points are being listed here. So this is what is in our control. We've got patient information, apheresis information, a lot of manufacturing information, final product, which is very key, cellular kinetics, and PK/PD. What do we do with this? And that's where the AI comes into play. And I think the next slide will talk about that. How do we group these models so we can at least try to get some kind of answers? Now, granted to say there are a lot of questions, but we wanted to focus in on 2 or 3 basic questions for the very reason that cell and gene is really burgeoning right now. We don't have a good understanding of the cell and gene manufacturing process. So let's look at simple questions and see if we could answer those. So the mission is on the left-hand side, and the questions that from a research perspective we wanted to explore were, can we have a better understanding of what are factors that are important in manufacturing? Could we use these then to predict any outcome, any clinical outcome? And what do we learn from our initial explorations that can then be translated into the next generation clinical trials or other clinical trials? So how can we translate this learning, and how quickly can we share it with the other clinical trials? That being the overall picture, we're just going to go through very quickly. On question 1 was understanding factors that are important to manufacturing. Question 2, how can we predict? So how did we combine our data sets? We had one set of data that convalesced into the patient group, next to the apheresis group, then the manufacturing data set, and then ultimately the product. And the question is, how can we understand what will make a good product? And that's why the product over here is outlined in red. Similarly, we know this is not the end, and we know based upon our discussion so far, there's lots of other factors that can be confounding. So now we increase the number of influencing factors on the right-hand side. I think, are we on time? There's, I think 1 more minute. So now we have patients, manufacturing, dose, and response, and we want to see how do these, the dose and the final product characteristics have an impact on response, knowing that the other factors also will have an impact. This is how we are grouping our information. This is how we are getting our initial data. And this graph is basically explaining what could be our confounders between dose and response. So I don't think I need to explain this in large detail, especially when I don't have the time. And basically, we are trying to group the data, apply machine learning models, learn what we've done from random forest analysis, and basically try to build a system of intelligence where we are ingesting the data, we are learning from the data, apply both statistics, but machine learning and cloud computing, and then figure out after ingesting this data, what are the insights that are coming out? How can we validate these insights and then try to generate a report? So we are in the very early stages of all of this, but it's a very exciting stage that we are in. So a lot of work going on and a lot of people working on this for about, a few months to a year now. So with this, I really want to thank the Forum for Regenerative Medicine to give us this opportunity to have a discussion with you, and also the Novartis teams and our partners who have been working on this. So with that, I'll just go back to the slide and stop.